So, welcome back. We shall continue to look at how to optimize drug target interactions. So, just to recap what we have been looking at in the past few lectures. So, we have sort of defined what structure activity relationships are, and these are basically the way in which we want to modify the structure in order to improve the activity of the molecule. And we have looked at various functional groups such as alcohols, amines, esters and so on, and how they can interact with the binding site by hydrogen bonding and by other weak interactions. And we have looked at full cations and anions which can interact by ionic bonding. So, some of the other major functional groups which can interact in this manner are the alkyl, aryl and halogen substituted compounds. And we have also seen that halogen substituted compounds may have a propensity to do alkylation, which is not very desirable. So, the relevance of the functional group to binding can be understood when we make analogues. Okay. So, here what we do is we either modify or remove the functional group altogether and see whether the activity is affected by such a change. So, we have seen all of these concepts in the past few lectures. So, we also introduced the concept of an isostere, which is basically a group that has the same valency. So, an isostere for an alcohol would be CH2, NH2 and so on. So, for example, when we want to interrogate whether a particular property such as hydrogen bonding is important, we would replace that molecule with an isostere which does not change the molecular weight a whole lot, but would change the polarity and would change the hydrogen bonding capability and so on. And we also understood why in vitro testing procedures are important, because once we start looking at animal models, the drug may actually not reach the target and that may be difficult to interpret. So, then finally, we were exposed to this concept of a pharmacophore, which basically summarizes the group which is important for binding. And we also looked at the concept of a 3D pharmacophore, where you can sort of draw triangles which relate these various functional groups. So, the need for optimization is very important for us to understand, because once we discover a lead, maybe through serendipity or through some screening or through some rational design, that lead compound may have good activity, but they are far from ideal. Okay. So, some of the lead compounds although have activity, but they may be low, and so you need to improve it. And one of the major problems that we face during drug target optimization is poor selectivity, and this can also lead to significant side effects. So, the lead compound may also be difficult to synthesize, and so there is an advantage in finding analogues with improved properties. So, we have seen thus far a number of strategies that one can employ to optimize the interactions of a drug with its target and to impart some level of selectivity. So, today we shall continue with this approach, and we shall look at various aspects of trying to optimize these interactions. So, the first concept that we will look at today is chain extension contraction. So, let us imagine that a drug interacts with a particular binding region or a binding site, where there are two different binding regions that are present. Okay. So, here is the first one and here is the second one. Okay. Now, if you imagine that there is a drug which binds to this, or your lead compound binds to this. So, it is possible that one of the regions that is bound is maybe strong, let us say this is quite strong, whereas this region is relatively weakly bound. So, by extending the chain, what we may be doing is to increase the number of interactions that may occur. So, this is one of the strategies that is frequently employed to improve binding to the particular region that of interest. As a corollary, one can also think about a situation where the chain is too long, and so you have some weak interactions that are happening over here, but once you contract the chain or reduce the length of the chain, then it is possible to arrive at a more optimal interaction. So, what we to think about here is that based on the pharmacophore that we have, there may be one binding interaction that is important, and there may be another binding interaction which is important. And so, now by modulating the chain length here, we can increase or optimize these features. So, therefore, shortening or lengthening of the chain is a useful tactic to try. The next concept that we shall look at is ring expansion or contraction. So, again here we are going to look at this example where we have couple of binding regions in this binding site, and this is the drug that we have with us, or the lead compound that we have with us. And here there is a 6 membered ring, and so it has a particular orientation that is going to pretty much rigid. So there is a hydrophobic region that is present in the binding pocket in which there is a weak interaction. Okay. So one strategy that we could employ is to improve this binding by changing the ring size in order to improve the binding efficacy. So when we move from a 5-membered ring to a 6-membered ring, what we are doing here is we are changing the orientation in which the ring interacts with this hydrophobic region so that it improves the binding efficacy. So therefore, this might be a good strategy to employ. So one example that we can look at here is in the case of this drug, which has weak interactions. As shown here, there is a carboxylate in this molecule and then there is a carbonyl, and there are weak interactions that happen. Now there is another binding region over here which the carboxylate, although it is binding, it is not very close enough for it to have effective binding. But once we change the size of this ring and make this a 7-membered ring, then what happens is that the molecule is able to access this pocket and it increases the strength of the binding. Okay. So this is an example wherein you can use this strategy to your benefit. Another popular strategy which is used in order to optimize the drug target interactions is to vary the ring. So for example, a benzene ring may be important part of your drug. Now the benzene ring can be converted to a heteroaromatic ring, or we can put in a substituent on the benzene ring, for example, and this is a very popular method to vary the drug target interaction. So there are a number of non-steroidal inflammatory drugs which have this 1,2 biaryl system which is attached to a central ring. So we shall look at these examples. So here then various pharmaceutical companies have tried to make various analogues in order to optimize the drug target interaction. So here are a couple of examples over here where this kind of a strategy has been employed, and another set of pharmaceutical companies had a different approach, and here they used a benzene ring and they used this thiophene ring, for example, to optimize the interactions. So a lot of these changes have been carried out in order to avoid patent restrictions. So keep in mind that patent protection is available for most new drugs, and in order for other companies to overcome this, what they do is that they try to make significant changes to the ring or to a particular area of the molecule so that they will be able to avoid the patent restriction. But some of them do indeed result in significant improvements in activity, and these are termed as me-better drugs. So the example that we can look at here is the antifungal agent which acts against an enzyme that is present in both fungal and human cells. So what was found was that replacing this imidazole ring with a triazole ring results in better selectivity against the fungal form of the enzyme when compared with the human form of the enzyme. So here is a case where the rest of the molecule pretty much remains the same, but by changing a particular functional group or a ring, we are able to achieve better selectivity. So one advantage of altering an aromatic ring to a heteroaromatic ring is that it also introduces the possibility of extra hydrogen bonding interactions. So herein what we can combine here is the strategy of changing the ring size or changing the ring along with the extension strategy that we have looked at previously. So we not only want to make a change in the binding affinity of the benzene ring, let's say, as compared to the heteroaromatic ring, but we also want to increase the number of possible interactions through the extension strategy. So here is the example that we will look at. So the structure 1 was found to be the lead compound. It's a tricyclic molecule with one pyridine ring and one benzene ring which are connected by a heterocyclic ring. Here what was suggested was that we convert this benzene ring to a pyridine ring. So the pyridine ring, what the advantage of having a pyridine ring is that it actually can have an additional binding group, because that pyridine ring has a lone pair which can interact through hydrogen bonding perhaps. So this molecule was now the new lead compound, and using this molecule the researchers ended up with this nevirapine, which is an antiviral agent. The next method that is commonly used is called as a ring fusion strategy. So here what happens is that when there is a possibility of fusing a ring or creating a new ring along with an existing ring, then that can result in a different set of interactions. So this may prove to be useful in not just increased interactions but also increased selectivity. So we already looked at previously that there are molecules such as beta blockers which are very useful, and these molecules have been developed as replacements of the aromatic ring in adrenaline with a naphthalene ring. So, here this is the molecule that we are looking at, and what happens is that this compound was able to distinguish between two very similar receptors, which is the alpha and the beta receptors for adrenaline. So, one of the ways in which we can explain this result is that by doing a ring fusion, we are actually introducing a new substituent for additional van der Waals interactions. So, the beta receptor has a larger van der Waals binding area compared to the alpha receptor, and so it is possible that this binds better and it improves the selectivity. Another possibility is we have looked at previously, which is what is known as steric blocking, is that the size of the receptor can be different. So, the alpha and beta receptors are going to pick up adrenaline, and so they are optimized in that size. But when we develop this drug, it is possible that you may have some significant steric hindrance to binding. So, both these concepts are possible, may be operational in this case.